Hello and welcome to Nikolai's genetics lessons, and here's a problem. A zygote with a genotype as follows will produce how many different chromosome combinations in the following cell types. And here different cell types. Basically what zygote means, this is fertilized egg cell. And if this egg cell would develop, of course would have all this tissue that is listed here. And we can separate this list to two types of tissues. One tissue would be somatic tissues, where basically all cells would be replicated by mitosis. For example, this can be eye cell, where mother cell would make a copy of itself, would make two cells, and those cells would be identical. The same we see here in liver cell and brain cell. So one mother cell would produce two daughter cells that is going to be genetically identical. So only one variant here possible, one variant here, and one variant here. But with egg cell, different situation. Egg cell would be created by meiosis, and during meiosis we would see recombination. We would see crossing over between two chromosomes, homologous chromosomes. So let's analyze what we see here. First of all, how many genes we see here. Some people may think that here is one gene, second gene, and third gene with the name CD. And this would be the first place where you can make a mistake. Actually, we have here gene A that represented by two alleles, A plus and A. We have gene B also represented by two alleles, B and B, so this is homozygous condition. And here we have two genes, C and D. Each gene represented by allele C and C plus, and gene D represented by two alleles D and D plus. But why strange notation that is different from what we see here? I will explain in a couple minutes. So let's first find how many different variants of the gametes we would have if we would have this problem simplified to only one gene. Imagine that here we have one chromosome inherited from the mother side, another chromosome inherited from the father side, diploid organism for the gene A. So say here is position or locus of the gene A would have two different alleles. On one chromosome we may have allele A plus. On the other chromosome that is inherited from the father side, we may have different allele, allele A. This is still going to be the same gene, but slightly different versions of the same gene. So in diploid organism, we may see a situation when on both chromosomes we would have, say, A plus, or A, and we can see A plus and A. So three combinations possible if the gene pool consists of only two alleles. So, how many different gametes such organism can produce? And of course it is super easy to say that one variant would be genotype A plus in the gamete. So what kind of gametes this organism would produce? If we are told that this organism would produce X cell, that means that genotype of this person, except what we already know here, is X and X chromosome. So this is female, because males do not produce X cells. Males produce sperm. So this is female with such genotype, and if this female would have only one chromosome, such female would be able to produce two types of gametes. One would have allele A plus, and another type of gamete would have allele which is going to be A. So also would be XL, because this is females that produce X. Now let's think how different combinations would affect our calculation. And imagine that here we have, so this is chromosome number 1. Now we have another chromosome number 2, also represented by two different chromosomes, but homologous chromosomes, one inherited from the mother side, another from the father side. So let's say this is chromosome number 2. And here we have two alleles. Both of them are small b and small b. So this is the same type of alleles. Would it create new variation in the gametes? Actually not, because one chromosome would go here, another would go here, and no matter if this would end up here and this would end up instead of here, it doesn't affect genotype. We still would have here small b and small b here. So, because for the gene B, this organism is not heterozygous but homozygous, it doesn't create new variants. So we basically can ignore such variants. It doesn't affect our calculations. I just show you here in order to demonstrate that when we have a homozygous situation for any locus, it doesn't affect our calculations. We just can ignore it, cross out, and move to think about other different variants. And this variant is slightly different from our previous two variants. How we should understand it? Basically, we have, as I said, two genes here. Once again, imagine that we have here chromosome number 3. Also, we would have two homologous chromosomes, one inherited from mother side, another inherited from the father side. But this time, on this chromosome, we have two genes. So this notation means two genes on the same chromosome, and this would be on the different chromosomes. So we have here 3 chromosomes, and we have 1, 2, 3, 4 genes. So we have once again somewhere, say here, locus for the gene C, and this gene is represented by two alleles, allele C and C plus. Also on the same chromosome we have another location of the gene D, or locus, and this gene also represented by two different alleles, allele D and D plus. So how many different variants we may have here? Because XL is result of the meiosis between these two homologous chromosomes, we can see crossing over, and this crossing over would create new variants of the gametes. So we may find a first variant of the gamete C and D, this chromosome, without crossing over, or if crossing over would happen 2 times here between these two genes, basically first it would create a new variant and then would reverse back. So one of the type of the variants we may have here is CD. This is going to be parental genotype. Another would be C plus and D plus. And we may have also recombinance as a result of the crossing over. If crossing over would happen once, we would see recombinance as C and D plus, and C plus and D. I forgot to put plus here. This is going to be here parental genotypes if no recombination would happen, and these genotypes would be if recombination would happen, if crossing over would happen once or 3 times or 5 times. So when it happens odd number, we would have recombinance. If number of crossing over would be even number, we would have the same still parental genotypes. And of course the distance between these two genes affect how often this crossing over would happen. If two genes would be on the opposite sides of the chromosome, we would see that probably in 50% we would see parental genotypes, and 50% we would see new genotypes that is result of crossing over, so we would see recombinance. But if two genes would be super close to each other, maybe we wouldn't see any recombination at all. So let's assume that there is some distance between these genes, and crossing over may happen between these two genes. So now we have found that this chromosome number 3 can produce 4 different genotypes. So parental genotypes and recombinant genotypes. So now we are ready for our final stage of calculation. So how many different gametes we can see here with different genotypes. And here would be, so let me use a different color. So this would be one combination, and this is going to be A plus B and CD. And here would be second combination, that is going to be A plus B, C plus D plus, and this is going to be third variant and fourth variant. So 4 variants here, and also we may have different combination. So A, B, C, D. We may also have such variant as A, B, C plus D plus, and this is going to be third and fourth. So basically, as you see, we have 4 variants here, 4, and 4 variants here. So total number of different variants of the genotypes of the egg cell would be 8 variants. Let me list at least a couple of variants to demonstrate. So once again, here we have genotype A plus B, and if it would combine with this genotype, we would have A plus B and C, D. So this is going to be genotype of the egg cell, and another variant would be the same, A plus B. So you have to understand that A plus stands for this chromosome, B stands for any of these chromosomes. We still would find B allele here, and here we have also chromosome with a CD as would be parental genotype, or C plus D plus, also parental genotype. So instead of these letters, we can put just chromosomes here. So gametes with different variants of the chromosomes. And C plus D plus would be second variant. And we may also have A plus B and recombinant, that is going to be C, D plus. So this is going to be third variant of the gamete, and another one would be AB and C plus D. A plus B and C plus D. So fourth variant of the gamete. And the same we can do once again with all these 4 variants. So we are going to get 4 variants of the genotypes here. So AB would combine with CD. So AB with CD, and another variant AB with C plus D plus. So AB with C plus D plus, and we may also have A, B, and C and D plus, third variant. So this is going to be this variant. And the last variant would be AB would combine with C plus and D. So as you see, 8 variants of the X cells is possible, 8 variants of the genotypes, and of course I could explain this problem within 1 or 2 minutes, just giving you a mathematical variant of the solution, but I wanted you to understand in deep, in every small detail, how this happens. So this took me a little bit longer than usual, but now I hope you would be able to solve analogous problems. You wouldn't have problems understand what this notation means and how many variants in the X cell we may get when we have 3 alleles. I hope now you wouldn't spend your time thinking about how homozygous alleles would affect our calculations, whether it is going to be homozygous recessive as in our example or homozygous dominant. We can just ignore, we can cross out these variants here, and if you next time want just mathematical method of solution of this problem, would be easy. So we have one heterozygote, second allelic pair that is heterozygote, and third allelic pair that is also heterozygote. So we have 3 heterozygotes here, and basically it is very easy to solve this problem mathematically. Everything we have to find out how many genes are heterozygous. We have to ignore those that is homozygous. So if we would have one gene that is heterozygous, this would produce 2 variants of the gametes. If we would have another gene, so if we would have 2 genes that is heterozygous, say this combination here is represent situation capital A small a. Second variant represent 2 genes capital A small a and capital B small b. So such genotype would produce 4 variants of the gametes. And genotype that is, for example, capital A small a, capital B small b, and capital C small c, how do you think how many variants it would produce? We already discussed this situation here. It would produce 8 variants. Variants of the gamete, so 3 genes would produce 8 variants. And now you see the pattern. 1, 2, 3. This is number of chromosomes that is heterozygous. And here we have another 2, 4, 8. So as you see, with each new heterozygous chromosome, this number doubles. So now you can answer such a question. For example, if we would have 4 genes that is going to be heterozygous, how many different combinations of genotypes we would see in the gametes? And you can say that now this number would double. So we would have 16 here, 16 combinations, and 5 genes that is heterozygous would produce 32 combinations. So as you see, we can solve this problem mathematically under just 1 minute. If you would have such problem on your exam, just write down this logical pathway: 1, 2, 2, 4, 3, 8, 4, 16, and 5, 32, and so on, and you can solve any analogous problems within seconds. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.